forgot to say I agree on the other screen. Continue and come back here. Okay, so this is gonna be day 3 of the advanced Cytoscape class or workshop. We'll pick up wherever we left, but if you can upload the current version of the day 2 script, then you'll be in sync with whatever I've added to the end of this file to finish up what we've been doing yesterday. So I did put the link to day 2 drive with this file present. So you should be fine. And then yeah, so that's the current version of the spider file, and we're gonna, as I said, pick up where we left before, uh, except that instead of playing with some toy version of XML file that I used for the examples yesterday, we'll try to build one of the files, or improve one of the files that I used on Tuesday to get our network into Cytoscape. Just to look at the original file, I'm gonna open another file from the Google Drive. It's gonna be the new file. Not this one. It's gonna be the get filtered node version 2. This is the one that we're gonna use. Okay. This file is nearly exactly identical to whatever was used on Tuesday. The only difference is, instead of in first column using the official gene or locus links or whatever names for the individual genes, I substituted those with Uniprot identifiers for corresponding protein. So this is, otherwise the file is the same. There's a, in first column there's identifier for a protein, and then there are 6 columns corresponding to expression levels and reliability for 3 separate conditions. Okay, so this was used originally, or the corresponding original file was used to annotate the cytoscape graph, and it was used to it was used to fill up the node table, and then the information in this table was rendered in the graph. In the table in the cytoscape that you see here, we've got 2 additional columns, one with name, the formal name. Well, there's actually one additional column here, the one with the gene name. Okay, and this column came from a separate file that was also provided on Tuesday, and now what we're gonna do is we're gonna actually get after those gene names and a few other things, not by contacting Uniprot and getting records from there, and then pulling information out of there and putting it into updated version of the file here. So that's our initial goal. How to do it? Well, first thing to notice is, as I mentioned, I think yesterday, is that once you go to Uniprot, let me see if I got it somewhere. No, I don't. So let's go to Uniprot. Okay. So if you've got an entry here, let's say gal 80 list. Okay. Once we find an entry for the gal 80 protein, when you use the browser, the links end up with the identifier for the protein entry. If we add .xml, then the server returns the XML version of the page with all the data there. So we're gonna pull files that look like that directly from the server, and then we're gonna extract information we're interested in. What we're gonna be looking for is we're gonna be looking for the gene name. That's the one that shows up here. Let me see if I can make it bigger. Can I make it bigger? No, I can't. So this is one element in the XML file that we're gonna go after. Okay, this will let us generate the human readable file in the network. We're gonna get the formal name, the so-called ordered locus. This is what we used before as an identifier. So now our input file doesn't have it, so we'll pull it in as well. Doesn't hurt, and it doesn't hurt to get the species for each protein. So we're also gonna pull in a couple of elements from the organism here. So there's many other things that can be put in. Whatever you see on the web page is visible. It's available through the XML. So if there's a need to pull in some other piece of information, it's the same procedure. It's just going after different elements in the file. But the first thing is to get the files. So let's see how to do it. So here's my file. No, this is not the one. I want to go file open. So this is day 2 and the spider script here. Let's open it. Okay, so if I scroll down, the last section, last cell in the file is gonna correspond to whatever we've got to do. Okay, so I'm starting with importing a couple of standard modules to get access to different functionalities that I'm gonna use later on. But otherwise, I do specify the file that I'm gonna start with. This is the file that I've shown you here. It's also on Google Drive. This is the one with the first column corresponding to protein identifiers and then expression data. So this is our starting file. This is what comes out of whatever experiment you might perform or you might be asked to visualize in Cytoscape. This is coming from people doing experiments. So they typically would provide a list of genes or a list of proteins and some experimental data to deal with or analyze. So this is coming from someone doing experiments. So we're gonna start with this file and try to get some extra information out of Uniprot to annotate the individual proteins there. So that's our initial file. Okay. Now I do have another variable keeping the name of the file that I'm gonna be writing the results to, so that I'll retain the original file as is, and then there's gonna be another copy of the file with the information I need. Okay. Now, if you look at Uniprot Records in a Cytoscape, I mean in a browser, you're gonna notice that if you don't see it here, you've got to go and view, uh, and you've got to view source. I think Chrome is showing up source by default. Here it's sort of formatted, so I'll switch to view page source, and then the plus, let's see, yeah, that's better. And then what you're gonna notice is that the top element here does have a schema defined. It doesn't have a prefix, but still all the elements are coming from so-called default schema, which is the schema used without prefix, and that's the one specified here. So we'll need this information to be able to access individual elements, to specify individual elements in our XPath expression. So what I did is I just took the name of the schema out of here and created myself a dictionary here with the prefix I'm gonna use in the XPath and corresponding schema down here. So this is the line here. Then I need the place we'll be looking for the files, for the records. So this is the part of the URL. This is part of the location where the files are showing up. This is taken from the URL that shows up in the browser. I just took the everything except for the protein accession protein identifier and put it into variable here. This way, once I append the identifier and then .xml to my URL, I'm gonna have a full path leading to whatever file, whatever record I want to download. So this is my second line, and then I'm gonna put all the files that are read from the server into a directory that I created in the directory where the script is. This is just to be able to keep track of what I'm getting. It's first, and there are two reasons to do it this way. One reason is getting files from over the network takes time. So it's reasonable to get a file once and then store it locally so that the next time you need it, it's already available without waiting for the server to respond. So one reason is to speed things up. Second reason is it helps documenting what you're doing. When you're clicking in a Cytoscape, when you're clicking on the browser and generating different files, different things, when you repeat the procedure second time in a week or a month or half a year, you're not guaranteed that they're gonna get the same result because the sites that you're contacting updated information. The Uniprot records, for example, are updated quite frequently. You can check it by looking at the history for a given record. I went back to the human readable version of the record, and then when I go to history and check previous versions, come on, you're gonna see that there's a lot of versions showing up there. They are updated, as you can see, every couple of months or so. So if you go back half a year, someone's generating me background noise. Let me eradicate this thing. Oh, maybe not. No, okay. Oh no, I'm gonna eradicate the noise. Sorry. I'm gonna mute. Yeah. Okay. So as you can see, the records in Uniprot are changing every couple of months. So half a year back, you're getting three versions behind, and you might be getting different version of the record. So in general, it helps to keep track what you're doing by storing the information that you're getting from outside somewhere on your local drive, so that you can have a exact information what was the input to all your on your analysis. And this is the second reason uh, to store the initial input file somewhere, just for documentation. So and then once you've got those input files and the script that does the processing, then you know exactly, you can reproduce exactly what was done. With with accessing things through the browser, one problem is every time you do it, it's different. And second part is you've got to record somewhere on paper what exactly was done in order to be able to reproduce things. So when you're accessing doing things on the web, problems with reproducibility in terms of keeping track of exactly what was done. So that's my location where the files were gonna end up in. Okay, so first we've got to get our input file, and we've got to go after the identifiers here. So that's what's going on. And actually the entire script is going one line of the input file at the time, gets the identifier from the Uniprot, then gets the record from the Uniprot, and then starts processing it. So here's the top level loop where we're opening our input file, this guy. Okay, and once it's open, we're also opening the file that the result will be written to. I'm using W for write as the mode for the open. This way, every time this file is opened, it's gonna be wiped out clean. So every time I run the script, the file will be erased and filled up with new information. If I were to keep adding things to the file, I can open it in append mode, and then the next time the script is run, the new information written to it would be added. So I'm using W because I want to start from scratch. But I've got to remember that in case I care about the file I've got and contents of the file, I've got to make a backup copy or something, do something, because otherwise the script might erase it. So that's my output file that's open, and then I'm going over the NF file. So this is my input file. So I'm getting one line from it at a time. I'm stripping white spaces in front of the line at the very end of the line. So that there's nothing hanging out here. There's no spaces here by chance. There's for sure there's a new line, so it's gonna be stripped by applying strip. And then I'm splitting it on white spaces. The split method applied to a string is gonna divide a line into separate fields, and this file is tab separated. So that's the default behavior of split. Depending on the file, if there are empty spaces or whatnot, one can provide, or commas or colons, or one can provide an argument here. The default is, I think, \t, which corresponds to a tab. So that's what's happening here. I'm just getting one line from the file, removing empty spaces, leading and trailing empty spaces, and new line, and then I'm splitting whatever is left on tabs. So now in NF call, I should have a list with the information coming from the file. Let's try it. Well, we can always say here sys exit, and this is a way to stop the script. And in this place, I'm just using it for a moment to see what's happening. For debugging or demonstration purposes, I'm gonna here do print nf call here. So we're gonna stop just at the very moment, and I'm gonna comment this thing out because I'm not gonna be writing anything for a moment. So let's see what happens. So here's my script that it's gonna just start, open the input file, read the line, split it into bits and pieces, and stop, hopefully. So let's see what happens. Let's run it. What's going on here? Let's run it. Okay. So we stopped where the sys exit is. It looks sort of ugly, but who cares? But just before we do have a printout of the NF call, and this is indeed a list because what we see is surrounded by square brackets, and then we've got several fields. The first one is our identifier, and then there are the values for the expression data. So what we want now, we want just the first element, which is the identifier. We don't care for a moment about the rest. So let's see what's going on. We take this first element of the list. So we take the identifier here, and here we're putting together the URL that corresponds to what was okay, come on, to what was here. So now we're generating address of a web page that looks like this. This is what's sitting. This is what's sitting in my app URL. Then I'm concatenating the identifier, and then I'm adding at the end .xml. So that's what's going on here, and we can check it once more. We can now move this thing down one line, print this time my file, and once we run it, sorry, I'm not generating URL moment. I'm generating for a moment, I'm generating the place where the file should end up. So this is my file location. So this is directory where the file is supposed to end up in. Okay. Here, then I'm adding a slash because I need a slash here. Then I've got the identifier, and then I've got the XML added. So this is where the file ultimately is gonna end up in. Okay. So now, if the file is already present, it means we've run the script and got the file in. So I've got to check it. To check it, I'm calling the method here. It's defined. This is something that Python provides for checking if a file exists, and the method here or the function here takes as argument the location of the file, and this is going to return true if the file is there, or it will return false if there's no such file in a place we're looking for. So now, depending on the result, if the file is not there, so this is the branch here, now I'm gonna make the URL. Okay, that's what I was supposed to do. Okay, then I'm gonna just print this URL just to see it showing up on the screen. And then I'm gonna open this URL and pass it to our parser, our XML parser. As I mentioned yesterday, parse can take either a file that's local, but it can also take URL and parse the file that's remote. It's gonna take more time because it involves uploading the file over the network, so it's slower, but it's gonna generate exactly the same data structure as when run on a local file. So here we're parsing this remote file, okay, and now once we've got it, we can open the location where it should end up in. This is the one which we checked here is at the moment empty. There's nothing there. So now we are opening a file corresponding to this location, and now we are writing there whatever the operation reverse to parsing returns. So we're converting our data structure back into string, and now we're writing it to the file. Okay, and now once we're done, we're closing the file that we written to, and that's what's happening when the file is not local. If the file is local, then instead of executing the series of commands here that get the file from outside, we are just printing out the location of the file, and then we're parsing the file directly as a local file. No matter what, once we're here in this place, the my app DOM will contain the representation of the record. It's either gonna be coming from the local file here, or it's gonna be coming from the file that was uploaded on the fly from the network. And somewhere in between, we made a local copy of whatever was uploaded. So next time the script runs, it will get you the local file. So the second time you're passing through, the script should run faster because it's gonna be getting the local files. So let's see what's going on. To demonstrate it, first I've got to remove the record from the directory because I think it's already there. So let's do it. Let's go to the directory I'm using. It's gonna be up here. This is my Uniprot thing, and I'm going after. No, I'm going after what? P53, P5389, P53, where is it? Let's get P50, 53. I think that's the one that I want to get rid of. So let's delete it. Now in the directory here, I've got every other file but this first one that I was looking for. So let's see what happens. I'm gonna print file for, so I'm gonna stop here. Let's see. So I want to do sys exit here. Okay. So now let's see what's happening. Just remove, just clear console. Okay, so this is the first pass, first time the script is run without the file, local file present. So now let's run it. Let's see what happens. We don't have ET. Okay, so we don't have this guy. Okay, so I don't have the module loaded for the parser. So let's fix it. Let's get those two things from here and place it at the top of the cell somewhere here. Okay. So now I do have ET. Before I didn't run the first cell in the file, so ET didn't get loaded. I also need URL open, so I'm getting the other module that would be missing here. So let's clear the console and let's rerun it once more. Control enter. Okay. So now what do we see here? We don't see anything here. Well, we see the thing that got printed is URL following URL. So we're printing the URL that was generated here. So this is location of the record on the Uniprot server. Okay. We now open it and pass to the parser, and then we're creating a file with the name like this that's gonna be local. So now when I rerun the script once more, if everything works fine, let's see if that's the case. Now I see as output file and location of the file. Well, it means that the procedure works. It means that the second time I passed through around through the script, the file was already in there. So instead of getting it from the network, I did get it locally. Okay, so that's what's going on, and we can check in a directory if that's the case. If we sort this thing by date, here the newest file is dated just now, and that's what got uploaded when the script was run first time, and then the second time the script ran, the file was accessed from a local drive. So that's how the script is supposed to be behaving. So that's this looks fine. So it looks like at the moment we are here, we do have our record loaded into the DOM structure, either from the network or locally. Either way, we do have something to work with. Okay, so so now we've got to get to the things there. So let's see. So I'm making myself the beginning of the XPath that I'm gonna be using over and over again. The reason is I want to go to different things here in the record, so it's always going to start with Uniprot and entry, and then I'm gonna go some more detailed stuff from inside, but my XPath is gonna be starting like this. Alternative version is I could use essentially I could essentially say entry XP is double is just double slash. Okay, and this means look for something inside of the XML structure, irrespective of how deeply it is nested. Depending on circumstances, you might use one way or the other. This full path is gonna be most likely a little bit faster, especially if the files are big, because in this case, the script's got to check, knows exactly what to look for at the top level. In this case, it's got to check every top level and every possible nesting. Specifying wildcard like this involves more work essentially. So depending on the circumstances, depending on the size of the file, you might use one way or the other depending on convenience and so forth. So I'm gonna stay with whatever I had before, but this is alternate. Okay. And now I'm building a series of XPath extracting different things. So here's one, here's another one, here is another one, here's another one. If we look at some of those, well, so let's see what's happening with the first one. Okay, so the first one is starting with Uniprot and entry, then we're adding up gene and name. So we're getting to Uniprot entry, then we're going one level down to gene. We can fold a bunch of things here. So top level is Uniprot, then we're going one level down to entry, then we're going one level down to gene. Okay, and there are many names possibly here. What I'm trying to go after is I'm trying to get this gal 80, which apparently is in a name element with an attribute set to primary. So that's what I'm doing here. I'm going to the name, and then I'm setting up a condition that I want to find every name element, but only if it's got an argument called type set to primary. So up to here, I'm selecting for this entire element. But I don't want the entire element, I want just the text sitting inside. So I'm adding the slash text, and this will return me the actual content of the actual text between the opening and closing tag. So this is what's going on here. And this presumably is gonna return me the standard name of the gene. Okay, so this is our XPath here. Next XPath is this is I'm going not after primary. I'm going after primary. I'm going here. Sorry, here. So this is the guy that I'm going after, specified in ngname XP. For the ordered locus, so this is for the standard name. I'm going after name element, but once more limiting the search to names with the type set to ordered locus. So this one should return me the contents of the name with the type ordered locus. So far we've got the GAU, we've got the YML, whatever, and then there's a chance that some genes are so new or so poorly characterized that they don't have a human readable name, that all they've got is the YML 051 whatever W that came from the people who sequenced the organism and generated the systematic name. This is completely new gene that's known only by the systematic name. No one did any experiments. So there's no official name. So I'm making sure that this is not the case. If it is, then the search for the official name will generate no output. It won't be found. I'm checking for that by looking what my XPath search is returning, and if there's nothing there, if there's just a list with no elements, then I'm just appending to it first thing that the search for the ordered locus returned. This way, I'm making sure that in the gname there's either a formal, either a human readable name, or as a backup the name of the ordered of the ordered locus. So here I'm protecting myself from having nothing in a gname, and this is the reason the search for those two things are reversed. I first want to look for the ordered locus because it's gonna be always there, and then I'm looking for the second one. If the second one is not present, I'm using the first name to fill in as a backup for the second result of the second search. So far we've got the formal name and we searched for it, and in OLN this is gonna be a list with whatever the XPath found. Then we've got search for the human readable thing, and once more there's gonna be a list with presumably one if it was found, or less, or nothing if it's missing. So we're putting in to make sure there's always something there. And then there are two more searches, one for the name of the organism. Okay, so this is just as before. We're getting to this time organism here. So we're going Uniprot entry. This is the starting part. We're going Uniprot entry organism name here. And then we've got the choice between scientific and common. So I want to always get the scientific version of the name. So I'm going after scientific, and here I'm gonna get the name of the species, plus if there's something about strain, I'm gonna get strain. So this is just as before. I'm going after organism name with a type set to scientific, and then I want to get text inside. This is something which humans read. Computers tend to read the formal identifier of the species that was assigned to each species by NCBI. So it's available here as DB reference to NCBI taxonomy, and it's sitting in the attribute. So my XPath is going to look a little bit different. Here, here it is. Here I'm going after organism, but now inside organism, I'm looking for DB reference, okay, for the element here. In case there might be more than one, I don't know if there is or not, but I'm just making sure I'm gonna get NCBI taxonomy, not something else by accident. So I'm limiting search to NCBI taxonomy type set to NCBI taxonomy. And now I don't want to get the text because there's no text for this element. There's only attribute named ID with the value. So I want to get to the value of the ID. So what I do is I go and specify the attribute as at and attribute name. And this is gonna return me the NCBI taxon ID essentially. Okay, let's see if it works. So let's now print what we found. Let's print OLN copy. Okay, let's print shucks. Let's print copy. Let's print OLN. Let's print copy. Let's print gname. Let's print tax name copy and v, and let's print tax ID copy and v. Okay, so and let's then do sys exit. And to convince myself that I'm in the right place, I'm gonna just print here a bunch of dashes, so I know what's in the output. Okay, so now I'm gonna run it once more. I'm putting the sys exit to stop execution after going through the first file. I don't want to go through all the entries. So this way I can look just at one at the first thing to see if it works. And once the script seems to be working for a first entry, then it can run for all the others. So let's see what's happening. Once more, I'll clean the console and I do Shift Enter. So here's my printout with the dashes here. And now I should have four lists printed out. This is because the XPath search returns always a list of values it found. So I'm gonna have 1, 2, 3, 4, four lists. The first one is supposed to have an official name. So here it is. Ordered locus is here. Second one should have a human readable name. Here it is. Then there should be taxon name, possibly with a strain. So here's the name I was going after, and here's taxon ID. So it's here. So we're nearly there. So once we've got the information we need, we want now to combine it with whatever came from this file, and I want to put those additional things in a second, third, and so forth column, and then put all the rest after that. So I want to insert essentially two columns here. So let's see how I need it. Well, I created a variable, okay, that is gonna have a new line, and this is sort of counterintuitive. I want to have several columns, several pieces of data, several strings. I want to put them together into one line that's separate where those values are separated by tabs. So the counterintuitive way of doing it, I can show you this thing in a separate block for a moment. Let's say we've got the list of things. Let's say we've got our list of things that's gonna be composed of A and B and C and D. And when I print LL, okay, then I'm getting the default way the list is printed, which is starting brackets, ending brackets, quotas surrounding the elements. So this is not what I want to put into a file. What I want to put into a file, I want to take a tab, okay, and use this tab to join join elements of my list. Okay, so now what this thing does is it takes one element at a time from the list. So it's gonna take A, add tab after it, takes next element, add tab after it, and so forth. So we're gonna have A tab, B tab, VC tab, D tab. So now if we run it here, this is what's returned. You can convince yourself it's a tab because it's one character here when I'm highlighting. So that's how you can join a list into a string with arbitrary separator. If I wanted to make a comma separated file, I would just put the comma here, and then when I run it, I would have a comma separated line. So that's what's going on here. I'm taking a tab, and then I'm joining a list that I made by concatenating. This is my first column from the input file. This is this guy. Then I'm taking my values that I found with XPath. I'm putting all of them into a list, but I'm taking just the first element from gname, first element from OLN, first. So I'm just taking first initial value, the first elements from each result, okay? And then at the very end, I'm adding the rest of the, I'm concatenating the rest of the input file. So this is the first element in a input line, and this is second all the way to the end. So this generates me a list of things to join, and then I'm joining it using tab as a separator. Once I've got this new line, I'm printing out the line here, and then I'm writing it to my output file, and write doesn't add the new line at the end, so I'm adding also a new line so that I'm generating one line at a time. Let's see how it works. Okay, I can move now. I can get rid of the things here. Let's keep it here. I can get rid of this thing. I'm printing a new line here. I'm printing this guy, and I'm going to do nnf close here because I want to for a moment close the file here, and then I do sys exit, and I'm gonna run it. Okay, let's run it. So now I'm gonna read one line from the input file. I'm gonna generate additional information. I'm gonna, then it's gonna end up in the output file, and then we're gonna close the output file and stop. So let's see what happens. Let's run it. Name is not defined, yes, because at the very beginning I just commented out the open statement for the output file. So I'm gonna uncomment it here. Sometimes error messages are your help. They are your saviors. They tell you what's wrong, and then you can go back and find out what's going on. So here let's rerun it once more. Okay, we can figure out that we did get our file from the local drive because we see file printed out here, and here is what the new line is gonna look like. So there's identifier, protein identifier, human recognizable gene name, formal name, species, taxon ID, and then the rest. And then if we look what's in the file, let's go up here. This is our file, and if we open it, now we've got one line and nothing else. When we rerun the script, okay, let's rerun it once more. Okay, we see everything the same here, and the file looks the same. This is because when it was opened for the second time, it was open. It's being opened with the W flag here. So the file is created from scratch. If I were to add things to the file, then I can set the mode to A. And now when I run script for the second time, let's run it. Now what I see is I see two lines here because the first one is coming from the first run of the script, and second one is from the second time the script was run with the A. I can run it for the third time, and lo and behold, I should see now three lines. So this way I can append to the file if I feel like it, but I don't. I want to create just this file in one shot. So this is not what we want to see. So I'm just gonna go back to W. Okay, and now when I run the script, we're gonna revert to the single line. Now, so far we're looking at one line, just one entry to see if the script behaves as we want it to do. So once we checked it on one entry, let's see if we can run it on everything. Okay, I'm gonna comment out this guy. I'm gonna now comment out our exit. And now I want to close the file, the output file, not within the loop, but I want to close it after the loop is done. So I'm just commenting it out here. So the difference between the statement here is it's executed every time we're passing through the for loop, and when it's here indented one less level, then we're closing the output file once the loop is done. So this is after all the entries were processed. So let's try to see what's going on when we run it. Okay, and here's the printout from the script. And now if we go to the file here, now we do have our file with an extra information here. So this is what was added by the script. All the remaining columns are coming from the initial input file. So here's an example how you can start with your input file with your initial list of genes or list of proteins, and then you can annotate, add additional information by querying Uniprot and adding information from there by querying by using XML query XPath to query the XML file. Some of those operations can be done by clicking. Some of those operations can be done by using the web interface provided by Uniprot. The reason I'm doing it and prefer doing it this way is, as I mentioned, twofold. One thing is I do know what was exactly done because I do have a script that I can save, and then I know exactly what happened. And second thing is because I did generate a directory here with all the files that were used for adding annotation, I know exactly what those files are. And once you check inside, that's wrong. I didn't want to start it. Go away. Cancel. Go away. Go away. If I check one of those files, I can open it with this guy. Let's say those files are time-stamped. Okay, they do have information when they were last modified and what was the version. So I do have exactly the input data that was used for the analyzer or annotation or whatever. So this is the second reason. If I want to later on rerun this thing, let's say I want to rerun those scripts, the script in half a year or so to see if there's some extra information provided, I can always rename this database, this directory, to something like 2021040204. Okay, now I do have the directory with the files corresponding to analysis or processing done on the 4th of February. I can create new directory, new folder here called Uniprot. Okay, and now if I rerun the script, it will take time to go record after record, and it will fill up now the directory here with the currently up-to-date information. So I can rerun the script multiple times, each time specifying a new empty directory, and I'm gonna keep track of what's the data that went into analysis. So those are two reasons to do things this way, and that's why somehow I prefer doing it with a script and instead of clicking. To do it on Uniprot site, you can essentially go. You can essentially let's move it. You can essentially go to let's go this way. You can go to retrieve ID mapping, and then you can provide the list of identifiers here, specify what they are, specify what you want to. So let's say I've got identifier here. Let's make another one, P 60010. Let's have two of those. Okay, I'm gonna select, those are Uniprot identifiers. So I got Uniprot ID options. Let's say I want to go to, let's see, ordered, well, let's see something, let's go after something simple. Let's get, where's protein, no chemistry, protein, no PTM, bioprotocols. Let's get gene ID, whatever it is. Let's submit. Okay, and now we're getting one column with the initial identifiers, additional column with whatever we looked for, and this can be downloaded as a table. So there's a way to do this thing by clicking. The problem is once you don't know what contributed to the results, because you don't know what were the records used for generating the additional information. The second thing is you've got to document somewhere on a piece of paper what exactly options you choose for doing it. So some of those operations can be done by clicking, but once more, for at least a couple of reasons, I prefer to do it with a script. And this is a very generic framework of a script that you can modify to pull in any type of information from a Uniprot record, and then use for annotating things. So that's introduction to how to deal with XML files and records from Uniprot. And this is useful for generating input data for Cytoscape. So let's stop here for a moment. Let's make a 5-minute break, and then we'll go with the other file with getting to the network. So let's break it. Any questions? Okay, it's quiet, so let's make a 3 minutes break. I need something to drink. Sadly, what's happening here? Here. Okay, so use my mouse. Oh, all right. Okay, so let's go on with actually getting the network. We'll see how it goes. So that was the end of the day two script. Okay, so now I'm gonna get the script that is prepared for today. So this is gonna be script that's sitting in on a Google Drive in day three. So that's that's the one that you see. Oh, winter, no, they no. What did I do? It should be here. It's not here. Oh, that's strange. Where did I put it? I didn't put it there. Okay, so let me fix it. Let's get myself the script here. So here's day three at Cyto and open. Okay, here. So here's the script. I paste in the link to it once more. Where is it? Okay, so here's the link to the directory where this file is sitting. So you should be able to grab it. So now we're going after the other file we need to prepare. This is the file that was here. What the heck? Day one. This is the file that I had here. Okay. This is the file that has identifier of one protein or gene, identifier of the other protein, and the interaction type. This is what I used to generate the connections that you see in the Cytoscape here. So each line corresponds to one of the edges you see here. Now, how to get it? Well, the way the authors prepared this file is they identified the set of those 300 or so genes that they saw changing expression in their experiments, and then they went after whatever databases they had at the time, and they created the list of interactions that you see here. In a case where you're given a list of genes, you'll have to redo the process. I've shown you how this thing is done using Cytoscape and the Psychic here. I did demonstrate that you can specify here a search term and then create a network based on whatever you're looking for. But this is, once more, this is done through clicking, so it's hard to keep track of what was done. You don't know exactly what came in, what was going on in between. So if one cares about those details, then it's probably better to actually go after original interaction records and then extract data out of there on your own. It's a little bit more tedious, involves scripting, but then you know exactly what's going on. You've got more control over what's happening. So that's what I'm gonna try to show you how to do. But we're gonna be talking to Psychic servers anyway. The reason is a convenient way to get initial information, what the records are, and then start from there. So I did run initially, and there's another script in a directory called pmid count. Okay, it does take does specify the input file with the nodes. This is the one that we started with. So this is once more this guy. Okay, and what the script does is it's creating two two URLs for each identifier found in the file. Okay, and then one identifier is corresponding to querying the psychic server for the number of records corresponding to a given variable for a given protein identifier, and then there's another query that is actually asking for a next 100 records starting from whatever is specified. So the script is getting blocks of, in our particular case, of 100 records corresponding to whatever the identifier is used for a used in a query. The generic syntax of how to create those URLs is provided by the server documentation. Let's see where it is. Okay, here is documentation of the psychic and how the servers work. Okay, the syntax of what can go into query does have its own name, does have its own specification. That's, I'm gonna paste the link here. Okay, so this is specifying how the queries should look like. There are a few examples. There's information what can be put into the query, and there's also documentation for the server itself. We can go to no, we can go let's see. Well, we can go, let me guess. Let's go to IMAX console. Let's try here. This is the website for the IMAX consortium. This is the website that keeps track of who's contributing data to IMAX, and I'm gonna paste you this thing here as well. Okay, and somewhere here there should be IMAX. I'm trying to get an IMAX registry, but no. Let's see. Click registry here. Yeah, that's what I was looking for. Okay, and here's a page and another link, another URL that points to different psychic servers that are available. What you see here is it's the original source that, let's see, that contributes to the list of the databases here. What happens when you start searching psychic, then Cytoscape is checking what's in a page what's provided here on this page, and then talks to each of those servers and interrogates each of those servers for the information about the given the query. What we're looking for the server for IMAX, and here's the entry. So then if I click on the REST example, I'm gonna get a example query. Okay, so I can just copy it and paste it here. And this is exactly, I just added S here, but this is exactly where I took the initial part of the URL for querying the IMAX psychic server. Okay, and then there's up to here the part that doesn't change, which provides an address of where the query should be provided, and then query is provided as the last element in URL here. So this is where query goes. Okay, and then after the question mark, there are additional options. Here, the options are format equals count, and this is returning the number of results that would be available, and the version here is gonna result in a options specifying that I want to start from 0th position and I want to get 100 things. If I then run it once more but specify 100 as a first result, I'm gonna get a block of another 100 records and so forth until I go up to maximum to last resort, last record. So so this is how the URLs are generated. I took them from the description of the site servers at the place that I showed you. If one is interested in a different server, let's say, if we don't want to go after IMAX, but you want to talk only to intact database, then if you click on the REST example, you're gonna have once more different URL. The difference is apparently instead of IMAX as we had before, now it says intact, and the rest looks the same. So this is a convenient way of finding different servers and finding how to specify in a program where whom to talk to. Some of them are like IMAX intact are providing experimental data. Some of them might provide functional predictions or different type of information. What's exactly provided, it's listed in the last column. If we look something like, let's say Gene Mania, which is at the moment down, we see that there are protein-protein interactions, but there are also predicted interactions or functional links, functional relationships between proteins. Other servers might provide different things. There's a database that provides information about interactions between small molecules and proteins and so forth. And those are different databases that provide access to their data in a standardized way. So we don't have to worry about the format. It's going to be the same over and over again. So that's where the URLs are coming from. Okay, and then so this is where the results are read. Okay. Now, initially, we're reading the number of records that's available. We can see here what's there. Let's try to go to IMAX REST example. Let's get we're asking for something like ID and then identifier. So let's make something like P60015, and we're passing as format equals count notch. Okay, so this is our first query that we're executing. When we run it, we're getting the number of records to expect. So that's this initial request. What's going on is you've seen this URL open before when reading Uniprot files. The difference is here we're just assigning what this thing is returning to a variable, which happens to have a method called the read associated with it, which is going to return exactly what you see here, just the file, the information returned by the server. And in our case, there's only one line with a number there. So this HTML is going to contain a number of records. So then what the script does, it's going over range of starting from 0 up to whatever is the number returned in the first query. It steps by 100, so this range statement is going to return a series of numbers, 0 and then 100 and then 200 and then 300. Okay, because the int HTML is, this is 371. This is where this is the max number of records. So we're gonna get the range going between 0 and 300, and then for each specific value returned by the range, we're gonna be asking for my PSQ. So this guy followed by the query. So this is our query. This is going to be once more protein identifier, and then max result is 100. We're asking for a block of 100 records, starting from the record either 0. So this is going to be first 100, then second 100, third 100, and then for 300 there are going to be only 71 records returned, and the loop is going to end. So each time we're passing through the outside loop, we're getting 100 lines, each with the information we're looking for. And what the information is, you can see it here. It's essentially a big table. Here's one line. It's very long, so it's folded, and there's a bunch of stuff there. The format of this thing is specified. Oh, that's good. In another place, let's go. I had the link here. That's bad. Do I have it here? Here maybe. Yeah, let's get. Here's a paper. I'm gonna provide you all those links, all the references, so you can track where the information is coming from. And what's returned is here. We're getting essentially a table, and there are going to be columns corresponding to whatever are the two interacting molecules, whatever whatever are the identifiers, whatever are alternative identifiers, whatever are aliases for those two guys, what the interaction detection method, publication for publication identifier, and this is what I'm after. This is what I'm after. I'm after 1, 2, 3, 4, 5, 6, 7, 8, ninth column. So it's gonna be a column with the index 8 that's gonna hold information where the publication, the record came from, what this publication is. And if I now look at the look at the output, sorry, I was to get here. So the eighth column is gonna be, it's hard to find, but microscopy, it's here. So I'm after a column, a value that looks like this. Okay. It's built out of three parts. They are separated by vertical bars. And one of them has a prefix PubMed, and this is the identifier of the PubMed identifier of the paper that this record came from. So I want to get to this number. To do this, first I've got to split the line on tabs. This is tab separated line, so I've got to split it on the tabs. I've got to get the column with the index 8 to get to this particular column. Then I've got to split it into pieces using vertical bar as a separator, and then I've got to get the number that follows PubMed. So that's what the script is essentially doing. Okay, so let's where I am here. I'm clicking on the wrong thing. So this was my initial query to get the number of things. Okay, then once I've got the number, I know how many times ask for a next block of 100 records. Okay, so I'm going over those blocks. I'm asking for one block at a time. The response from the server goes into response variable. Whatever is there is read into HTML variable as a string. Now I'm splitting this one big string that corresponds to entire file, entire piece of data returned by the server into lines by splitting on a new line. So now in lines I've got one record at a time. So now I can go through the lines. I'm checking if there are tabs there, just in case there's an extra empty line somewhere. So I don't want the program to crash. So I'm checking that I can actually split on tabs. So if there's a tab, it means I do have a line with the record. So now I'm splitting on the tabs. In a column with the index 8, I do have fields. I do have where is it? Nah, I do have, where is it here? I do have, oh, go away. I do have here fields separated by vertical bars. So now I'm splitting on vertical bars to have a list with PubMed something and DOI something and IMAX something. Okay, so now I'm going over this list. I'm checking which of the elements starts with PubMed, and then if I got one of those, then I'm replacing PubMed with empty string. So what all I'm left with is a PubMed ID. So I'm removing, replacing PubMed with nothing, and end up with a number. And then I start counting how many times this particular PubMed ID did show up so far. To do this thing is initially I set up myself a counter that was set up here, how just in a moment, and then the counter is actually a dictionary. It's gonna have as a key the PubMed ID. So every new PubMed ID is gonna result in additional key added to the dictionary, and then if the PubMed is in this dictionary, it means there's already one or more entries there. So I'm incrementing the count by one. If the PMID is not there, then I'm just creating a new counter for this particular PMID starting from one. So then new PMIDs will initialize the counters with one. The PMIDs that already showed up will increment things one at a time. And I'm going over all the lines, and then once I'm done with the block of 100 lines, I'm asking for another one to go through all the records that are there. And then once I'm done, I'm returning the counter. I do return because I put all this entire code in a function defined here. Okay, the reason is I want to run the same code for many identifiers. It's got to be run once for every protein ID that I've got on my list. So it's convenient to put all those repeated operations into one function. So here's a definition of a function that gets counts for a new protein identifier. Okay, and the same publications may show up for different proteins. So what I do is I'm passing as an argument to the function the current version of the counter. Initially it's going to be empty, but and if I don't provide argument to the function, it's going to be set as empty by default. But if I provide an argument with the as a dictionary with bunch of counters, then the PMID count function will keep adding to those counters. So that's what the function does, and it's called after it's defined. So it's sitting somewhere here, and here's what's actually happening. I'm getting my input file. So this is the one with the identifiers and expression data and so forth. This is exactly what we did before. I'm opening the file for reading. I'm going over the lines. You've seen this thing before in the previous script. I'm getting the identifier from the first column in each line of the input file. Now I'm calling my PMID count function. First argument is identifier. Second argument is current version of the counters that I set up here as empty. Okay, but it's empty only when I'm passing through the for loop for the first time. When I'm passing it as an argument for a second time, I'm returning the version that was updated by returning the updated version from the function. So this thing every time a new identifier is provided to PMID is gonna increment the counters to individual for individual papers. And then once I'm done, I'm just printing out done, and then I'm printing out the contents of dictionary. So I'm looping over the keys, and I'm printing out key and the value corresponding to the key. I'm not gonna run the script because it takes time, because it's going over about 300 proteins, and it's going through many records. So this thing is slow. You can try to run it on its own and be patient. It's gonna take probably 10 20 10-20 minutes to run. Okay, that's the price of getting the data off the network. It's not gonna be as fast as getting things done locally, but it's got to be done once to get the list of publications and list of and the counts how many times each publication showed up. The result of the script is what the script generated is here. It's another file that I placed in a day 3 dictionary. And here's what the script returned. It returned a column with the PMIDs. Those are identifiers of the papers. And then there's a number of times given publication contributed interaction records for the set of proteins we looked at. And you can see that there's a bunch of papers that provided hundreds, if not tens of hundreds of publications. I'm tens of hundreds of records to interaction records. Okay, so there's a group of those 14 papers here. Those are papers that are that report large-scale experiments. Those are papers. If you check one of those, let's check this one, for example. This is PMID, so we can go and copy and then look for this thing in PubMed. Let's see somewhere. Let's go somewhere here. Okay, and if we take this number, we've got the paper that might have reported gazillion of complexes there for the nucleosome, and gazillion of interactions there. So this is a lot of interactions coming in a single paper. Here's another one. Let's go here. Okay, let's try another one. Paste. No, that's there. Not this one. Copy and paste here. Okay. Here's another paper that reports a lot of interactions here. Those are probably affinity complexes with a lot of interactions there. Okay. I think each record corresponds to specific cross-linking. That's my guess. So here's another one. Let's look at another one. Let's try this one. Copy, and look for something here. Okay, here's a typical high throughput. This is a paper that goes after protein complexes in yeast in general by mass spec, and there's a lot of interactions reported there. So this is typical high throughput. The way to find those papers is to look for number of records that are found in the interaction database. So initially, I'm gonna skip those papers with a large number of interactions because they take time to process. So I'll skip those for the moment. But essentially, At this stage, one would go through the papers you've got here, especially those with large number of interaction records, to decide which of those should be taken into account, which of them not, and how to deal with the papers with large number of interactions reported. Uh, so that we're left with papers that report smaller number of interactions. They are smaller, so processing is faster, and they are more convenient for working with when writing the code. And then once we've got the code running, then we can decide we want to add some of the other guys. So now the script we're gonna be writing is gonna be looking only for publications with less than 100 arbitrary number of records. So I'm gonna go only after those papers here, and then I'd like to get all the interactions reported in each of those papers. Well, how to do it? Okay, so interaction records that are provided by IMEX consortium can be downloaded from the IMEX database. If we from the Intact database, if we go up, no, if we come on, if we go up to, let's go to this way. If we go to Intact database, okay. So once we go to Intact database and to download, then we can navigate to FTP directory and we can go for PSI 2.5 version of the record, okay, of the records. We're gonna end up on a FTP server that gets us access to PubMed to interaction records grouped by the paper they came from. So this is in a PMID directory. Okay, we go here. We're getting bunch of subdirectories, each corresponding to a year where a given publication was processed by the database. So if we click on 2001, there's going to be a lot of files corresponding each to interactions coming from a paper with a particular PubMed ID. So this is gonna be a file with interactions coming from publication with a PubMed ID 1008 so forth. And if we click on this thing and open it with let's say with Notepad for a change. Okay, we're gonna get the familiar XML file that I've shown you yesterday, that is gonna have description of interactions from this particular paper. So that's how to get to one of those MIF files, one of those XML files describing interactions. So this file is sort of more complicated than Uniprot. So to process it, one can write code getting at XML from the scratch. With Uniprot, it's easy. The structure of a file is relatively simple. With interactions, the structure of this file is much more complicated. So it takes a lot of typing to getting to information one is interested in. So we recently wrote a module for Python that actually takes care of getting information out, takes care of parsing the data from the MIF files. So let's see how this thing is done. Okay, so let's go to our day 3 script. Okay, so here's the script. Okay, and let's see what's there. Okay, so the script is taking as input the file that I've shown you before, this guy, that was generated by the script here. You can try to run it on your own, and then it should generate and it should print out once completed successfully. If at the very end, two columns like this that then can be saved into a file that the day3 Python is gonna take. So this is the input. Okay. As before, I will make a directory where the interaction files will be stored. This is the same reason as with the Uniprot. I want to keep track of which exactly version of records I was using and what files did I use, so I can reproduce this thing and report what I used. Okay, this is advantage over clicking with Cytoscape, because with Cytoscape you don't exactly know what came in, and it's very hard to reproduce it. If you rerun the same sequence of operations half a year later, you might get something different, and then the question is what happened. Did I click something else half a year ago, or did the data change? So this way I do know what's in used as my input. So this is where the files will be sitting in. Okay, and then I do specify what's the location of those files. Okay, and this is the URL you've seen here, starting from FTP going up to PMID. Okay, so this is what's gonna be the same for every file I want to get in. Okay, the tricky part is that the year here is the year when the file was when the records were curated, when the records were entered into the database. So for a given PMID, we don't really know if a record was created in 2001 or 2005 or 2017. There's no way to guess where when it happened. So we've got to check every directory until we find the one with the record, if present. So here's a starting and last year of the directories. As you see, the directories are in sequence starting from 2001. So actually this can be changed to 2001, so we don't look for something that doesn't exist. Okay, so the first directory to look through is 2001, the last one is 2020. There's nothing else. In half a year one might add and go up to 2021 and so forth. So this reflects the current set of range of directories that's gonna be checked. Okay, and now what the script does is it opens the input file, the PMID file. It splits each line in default way, and then we're gonna end up with two columns. The column with index 1 will have the count, so it will have the number here. The column with index 0 will have PMID. So we need the first column, the value in the first column to create URL, and then we're gonna create it only when the count provided in the second column is smaller than 100. If I decide that I want to make the cut higher, then I can change the number here. I can set up the condition here in a different way if I got the different criteria for accepting the papers. At this moment, I'm just going after papers with less than 100 records. So that's what the if statement does here. And now, if the count is smaller than the cutoff value, I'm just printing the PMID just to see which one I'm looking at. Then I'm making the path to the file, okay, but this is only so this is the place where the file will be stored in. This is where if found the file is gonna end up in. And then I'm going over the possible years. So I'm going over the range starting from 21. So I'm just using this 2001, referring to it as my year range with the index 0. So I'm going after the first element of the list here, and then I'm going up to 2020 to get to the last one. So this is actually going to go to 2020 to 19, because that's how range works. So this should be actually 21. Okay, so now the range here will start at 2001 and will go up to, but not equal to 2021. So it's gonna go up to 2020. And then for each value of the year, I'm first checking if the file is present. Okay, if the file is present, we don't have to get it from the server, because it's not needed. So I'm just ignoring that case. But if file is not there, then I'm waiting for a second so that the server doesn't get unhappy. It's a courtesy not to run scripts that are banging on the server in a very rapid pace. So I'm just slowing the script a little bit down, little bit. So I'm just telling the program to wait for a second. Essentially, that's what this thing is doing. Okay. Now I'm making the URL by combining this constant part of the URL. I'm concatenating it with the year, and then I'm adding a slash. This is gonna be the PubMed ID, and then a .xml. So this thing is creating a path that looks like we've got, for example, here for this file, it's creating cancel. It's creating a, I can show you this thing down here. It's creating a path that shows up at the bottom of the window. It reads PSI 25, PMID, then 2001, and PubMed ID. It is not guaranteed that the file is there, because it might be not created in 2001, but in 2005. So I'm just printing the current year that I'm looking at, and then I'm trying to actually read and parse this file. Okay, if the file exists, then the URL open is executing fine, and the parse is gonna parse the content of the file. Once it's parsed, I'm gonna write it back into the place that the file is supposed to be stored locally. This is analogous to whatever was done for the Uniprot. So this is a very, very similar approach. I'm checking if the file is local. If it's local, then I do nothing. If it's if it's not local, I'm trying to get it from the server and then save it. Okay, and then if file is found, then we're gonna be fine. If file is not there, the try except block is gonna result in just no operation performed whatsoever. So we're gonna go back to next year in the for loop. We're gonna check if the file is there. If it's there, it means the previous pass wrote the file locally. Okay, if it if the file is not there, we're gonna check next year and then next year until we're gonna get to the directory on the server where the file is, and then the file is gonna be saved locally. So once we're past this loop here, we are guaranteed to see the file on a local drive. So let's see if that's what happens. Let's see first what's in the directory that I'm gonna be using. So let's see what's here. Okay, so I'm in day 3 and I've got IMAX. I've got several files here from the previous run. So let's rename this directory to backup, let's say. Okay, let's make ourselves a new directory here called IMAX. Okay, so we've got directory waiting for the records, and let's try to run this guy. Okay, and let's see what happens. I'm not printing anything here. Let's see. And I'm gonna make myself a little bit of space here, and I'm gonna clear the console. Okay, so go away. Okay, so let's run it. Let's see if I have, no, I don't, so let's see what happens. Okay, so we're checking for the first PMID on the list, and you see we're printing out directories that the servers, every time a new block shows up, new year shows up, it means we're just printed here, and it was repeated all the way until 206. Apparently, the file was sitting in a 206 subdirectory, so then we found it and saved locally in on the local drive, and we can actually check it by looking what's here. Okay, and now we found the second file. Now we're looking for the third one, and this thing will keep going for a while. It's relatively slow because there is 1 second waiting time every query, so that we're not abusing the server. I tend to do those things so that the people running the server don't have to complain block us and so forth. So this initial pass is slow, but once the script is run one time, we're gonna have the directory filled up with files, and from now on we don't have to worry about talking to the server. It's gonna run quite rapidly, and we will not we will have a set of input files that we can store. And here's another one that we know that we used for further processing. I'll try to stop the script, just kill it with Control C, or I'll try to kill it here. Let's see. Sometimes it's not too responsive. stubbornly running. Sometimes scripts talking to the network don't stop no matter what you try. So we might have to be patient for a moment. Well, I can be brutal. I can, for example, try to change this thing to for a moment. So this should make the script very unhappy for the next protein. Let's see, because it's gonna try to open a directory that doesn't exist. Just a moment, and it will end. It's a good time for asking questions. If you got questions, please ask. We've got the script to die. Let's see. Die. Come on, die. No, doesn't want to die. Okay, now it died. Good. Okay, so now I'm gonna revert back. I'm gonna revert back to IMAX X. Okay. So now the directory with the files with the files is partially full. We've got 5 files in here. So let's see what happens to the script. Let's rerun it once more. Here you've seen that we are always looking for file in different directories, and finally we found one and saved it. If we now run the script once more, then the first 1, 2, 3, 4 files were found instantaneously because they are local, and then the script keeps going on trying to add more things. So let's try to stop it once more. I want to stop it because I already do have directory with all those files. Okay, good. So so if I now just rename this one to part, let's say, and then rename this one back to IMAX. So now I've got all the files present from the run a few hours before earlier. So then if I run script now, it's gonna why is this 895? Okay, there's one more missing maybe. Let's see. So this is gonna be probably curated in 2020, I think, because we don't have 9A7 9. Yeah, we don't have it. Sure. Okay, yeah. So there was one file missing. This one was missing. So now if we run it once more, now the files are there, so we can access them locally. Okay, so we do have a set of files with the interactions to work with. Okay, now in principle I can go after the myth DOM variable where the records are stored, and then I can try to write XPath expressions to go after individual proteins interactions and so forth. But this is sort of tedious. It's doable, but tedious. So we did write a library that tries to do it for you. The library can be found on PyPy, which I used to have a link somewhere here. No. Where is it? Got too many things. No. Okay, so let's find it. Let's look for PyPy. Okay. This is a site where a lot of additional modules for Python are, and I can go into whatever I added there, which is this Pimax thing. And if you view it, I'm gonna give you the link here. Okay. So here's the link to the Pimax module. There's a short description of what it can do, but before uh, before using it, we've got to get it installed locally. For command line version of Python that's run outside of Spider, outside of Anaconda, you would have to type on a command line in a terminal pip I think install Pimax. This is, I think, how you're getting the modules into installed in a Python, provided they are coming from PyPy. The pip command is talking to PyPy and getting modules. In case of running Python inside Spider, we've got to run the pip from inside the Spider. To do this, we've got to run pip with exclamation mark in front of it. It tells the Python to actually run the pip install as a command in a terminal. Okay, so let's try if I'm correct. If we run it, okay, now we've got a lot of information here about what other modules might be required by the Pimax, and then at the very end it doesn't complain. So we do have this thing installed. Actually installed it before, so just confirming that everything is present there. From now on, I can access the module. I can import modules provided by Pimax, and here is information what can be imported. We're interested in a Pimax MIF. This is a module that that allows us to read and process the interaction files. Okay, so let's do it. Okay, let's import Pimax MIF. Okay, here it is. And from now on, we've got access to whatever the module lets us to do. So let's see what it is. Okay. So the way it works, according to the short description, is we've got to first create an object that allows us to process MIF files. So that's here. Okay, and then we're gonna parse a MIF file which is in the version 254. That's what I'm using because the directory the files came from, if you check here, are specified as PSI25, and if you read description of what's on FTP server, the files sitting inside PSI25 are actually in this particular flavor of the interaction format. So that's what I'm using here. Alternative version is if newer version of the format is called MIF 300, and corresponding files in that format would be sitting in a PSI 30 directory on FTP server. So there is a choice between those two. I'm sticking with the older version for a moment. So that's what that's how to get to a file. Okay, so now I just got to specify what the file is. So this is gonna be my file. Okay, and I want to check it. Okay, I've got to end here. So this is, let's see, how am I doing with indentation? Okay, so this is looping over years, and I'm done. And then I want to do everything inside only for the files within the cutoff. So this is the right right place to be. This is how deep I want to get. Okay, and I want to try it for just one file at the time. So here I'm gonna put my sys exit here, so that we're gonna check just one file at the time. Okay, and let's see what happens. Okay, so let's do Control Enter. As out here, I'm providing a full path to the file. So I'm providing the location of the file on the local drive. So this is gonna be something sitting in a directory specified here. But in principle, you can put here, as with the LXML library, you can put here URL, and then you would parse file from a remote location. I'm just using local file because then I can speed things up. I don't have to upload the file every time I want to parse it. So I'm passing here the location of the local file. Let's see, try to run it. Okay. So now we just stopped running it. Nothing but happened. We printed out the file, and then we stopped. So presumably, once we parsed the file, we do have our record sitting in a rec variable. Okay, so let's see what can we do with it. Well, we can go to Pimax Wiki. This is where there's more information about the module. We can go here, and here's an example of what's what can be done with the script, and description, what different things you can do with the parsed file. Okay, what we can do is we can steal from here. We can just iterate over interactions that came in the file. We can print interaction type, and we can print how many proteins are there, or molecules are there provided for a given interaction. So let's try this. Okay, and we do. So let's see what happens here. Okay, and here. So now we're reading and parsing the file, and then once we've got it, we're going over interactions that came in, and we're printing out the type, and we're printing out the count. Let's see if anything came in. Well, yeah. So we've got now we've got first interaction. The type is association. Okay, we've got 14 proteins there. This is association. So if you remember, one of the proteins there would be likely annotated as a bait. The remaining 13 will be praise. Same thing here, except now we've got 3 proteins, so there's going to be 1 bait and 2 preys, and here's another case with 11 baits and with 1 bait and 10 prey. So that's what we've got here. Okay, what else we can do? We can go after other things. We can list actually those proteins. Okay, so we can loop over for a given interaction. We can loop over participants. Okay, so and then we're gonna get for those what's its name, what's the host it originally comes from, what's its type, and what's its sequence if we wish to do so. Let's say I don't, so let's just get those 3 things. Okay, so here, and we want to go for we want to get participants of the interacting molecule for our current interaction. So the outside loop, the top level loop, goes over interactions that came in a file. The nested loop goes over participants that are for a given interaction, and here we're printing out the labels for the participant, for the species the original protein came from, and what this thing is. So let's run it now. And here now we've got more things. Okay, so here let's get to something with same number of proteins. Okay, so here we've got interaction type is association. Once more, there are 3 participants, and yes, there's one corresponding one with the accession ESA1 hist, and then another one. All 3 of them are coming from yeast. All 3 of them are proteins. And then there's the last of 3 interactions. There are 11 proteins there, and we've got those things listed. So that's what we're getting from this one particular file. The web page here gives you information about more things that you can get out, and in principle you can go after every entry in an interaction records. This is a relatively straightforward way of finding information about what was the experiment type, or what was the experimental host, what were the features, and so forth. All the elements in the file are provided by different methods that are described here. They all start the calls. Always specify which interaction you're getting at. So you can use an index here to go after first, second, third, and so forth, starting from 0 interaction. Then for a given interaction, you can find information about the experiment. You can get for experiment, you can get the method, you can get the host where the interaction was, the experimental host information, and then for participants you can get information about the experimental role, you can get information about biological role, you can get after features. So this thing makes parsing interaction files much easier. In principle, the library also lets you create files from the information you provide. When you're just analyzing interactions coming from databases, doesn't matter, but you can actually also create files this way. Now, one more thing that's useful, I think. Let's see. Do I have it somewhere here? No. Where's the pointer? Let's go back here. Data methods. Yeah. One more thing that the library should allow you to do is we want to go after interactions that are pairwise. We want to have interactions that are just connecting two proteins. So instead of interaction between three proteins of the association type, we want to convert this thing into two pairwise interactions, one between the bait and the prey, and then another one with bait and another prey. It corresponds to conversion of association like this, just with three proteins, to a series of binaries. So that's the spoke expansion of association, and the library lets you do it in a pretty painless way. We can check here what the method is. Okay, we can now instead of printing out the participants, we can just get our interaction. Okay, interaction, and expand it according to spoke model. Okay, and then we can save the result. Somewhere. Let's see. Bin list. Okay, let's save it here, and then let's print our bin list. Okay, so now I'm just reading the file, I'm parsing it to generate the representation of the file, and then once I've got it, I'm going over interactions, I'm printing out type and number of participants, and then I'm trying to expand it according to spoke model, and then I'm printing whatever the expansion did. So let's see what happens. Let's run it. Okay, we've got a mouthful, but if we look carefully what came out, let's say this is our last interaction that's there. Okay, we do have association with 11 things, and now what we've got the list, because there are square brackets from here to here, and then each element of the list looks like a dictionary. Okay, and this is gonna be our interaction. So now let's see what's there. We know that we've got the list here, so we can do for I in bin list. So here we're gonna go over the list, and now we're gonna just print bin list with an in. We're gonna print I, let's say this way. So this should get us one thing printed at a time. Let's see what happens. Okay, so now it's printed in a more regular way. It's always one line per interaction. Okay, this is, if we check what's the type of it, it's type. This is gonna be interaction. This is interaction so that we can apply whatever methods are described here for the interaction. Go away. Okay, so for interaction we can get as we did before, participants somewhere here. This is for interaction, we can get participants. So let's try here. Let's type I. So this is, we're gonna print out interaction, and then for participant in I participants, we can try to print P. Okay, and let's see what happens now. Okay, so now what we've got here, we don't have much success. So let's try. This is pretty uninformative output because the short label doesn't exist, so it doesn't help us much. So what we can do is now we can try to get, well, we can try to print different things. Let's try to print exp roll and see what happens. Here, exp, let's see, it should have, oops, no, this one, it should have exp participant P, well, by roll, exp roll, oh, well, I think exp rolls like this. We need exp roll participant. Oh, a participant. That's interesting. It should have. What is it? Just. It's interesting. Should have roles. Interactions. Okay, let's. Roles explore. No, it should have participants. Should have explore. First only participant. Explore. It should have for P import print P and print P and print. Let's see. That's annoying. It should have. That's surprising. Okay, what else? Full name. What about full name? What about P? Full name. Full name. Okay. Okay. P. How come it doesn't? It should have. Okay, so that's something strange. Let's do record builder one more thing. Well, let's see if it works. Okay. Okay. Well, I don't think I'm gonna find it now, but that's so it's sort of disappointing. But okay, so anyway, we went through getting one more thing. Let's see. Um, Participants for let's try here. Let's comment this thing out. Now let's try for oh, about okay. Let's go back. Let's do it this way. Let's this way. Okay, and let's do p interactor and label. Let's try it this way. What happens? Okay, that's better. Okay, so here at least I did get the label of the interactor. There's gonna be well, so this is essentially enough. So okay, so let's do it this way. So what we can do it if now, so what we've got to do, okay, so let's think for a moment. So if the interaction is binary, so if there are two participants for a given interaction, we've got what we need. We do have the starting and ending protein for creation of an interaction. Okay, so let's check what we've got. So if our interaction participants, if the length here copy, if the length here is equal to 2, we know what to do. We know that we've got protein, let's say A, our prot A is interaction participants copy and V0, protein copy and here it's gonna be 1. So this is gonna be our protein, and then we can now just print, and we want to print prot A, we want to print pp, and we want to print prot B. Okay, so this is the case, at least initially, if okay, so this is the case where we've got exactly two proteins per interaction. So this is already binary interactions can be represented in Cytoscape. Now else, okay, otherwise if there's only one protein, it means it's a self interaction, so we don't care, so we can ignore it. But so we can only we care if the length is greater than 2. So L if length is greater than 2. Okay, if length is greater than 2, now we've got to expand. Okay, but now it's the way to expand is gonna differ. It's gonna differ depending on the interaction type, as we've seen, as we've seen, as we've seen here. For association, we want to do spoke expansion. I think I've got a better thing. This is the wrong one. This is not I want to look at. Let's try they here. This is what I was looking for. What is it? No, this is also not the one. File open. I want day 2. That's the one I wanted, isn't it? Oh yeah, that's better. I was after this slide. Okay, so for associations, I want to do spoke expansion to generate those physical interactions in binary power and binary interactions that are physical. For physical associations, I can do matrix and generate physical, and for direct, I can do matrix to generate direct. Okay, so I've got to choose between spoke or matrix depending on what's the interaction type. So let's do it. Okay, let's now check how to expand things. Okay, so this is our binary case. Here's our 3 or more case. So now we've got to check what's the interaction type. We did print it somewhere here. That's right. Here we're typing interaction type copy. So now we can test if interaction type label is, and we were getting association apparently as we had here. Let's see. Do I have it somewhere here? That's right. If we have association paste, now we want to expand according to spoke model. Okay. Okay. Else if. What are the other options? Well, we might have physical association copy. If it's physical association, then we've got to copy and expand according to matrix. Okay, and we might have another case when we've got direct. Okay, so if that's the case, we're still expanding as a matrix. Okay, copy and paste. Okay, so now we're handling expansion for the associations, physical associations and direct interactions. Okay, so once we're past passed our if-elif statement, well, we're gonna make bin list initially set to empty. Okay, so now we're guaranteed that bin list does have something inside. Okay, well, that bin list is a list. So if it's empty, we have something else happening. So we're not gonna get any expanded interactions. But if there's something there, we're gonna go over one interaction at a time. Okay, we're gonna go over participants there, and well, you don't have to go over. There should be two, because this is already expanded. So we can do exactly what we're doing here. We can take here and we can get interaction and participants here. Okay, and now we can print prot A and prot B, and we don't need this guy. Okay, I'm not gonna, I'll comment the print part here. I'm commenting this thing. I don't need it for a moment. I'll print the guy here. Is there anything? Okay, so let's see what happens. Okay, so now we can run this thing. Okay, and we should see, and we don't see the short labels because of some strange reason. They should show up. Something is messed up. It might be participants. A moment. Well, I was getting participant interaction. No, participant. I know what. It's got to be interaction participants, and I want to print interactor and interactor, and then label, I think. Yeah, participant interactor. I want to do exactly the thing I was doing here. Okay, copy, and here copy, and then here copy, and here. Okay, let's see what happens now. Here we go. Okay, there's one extra stuff being printed. Interaction. I'm getting this thing printed out in obnoxious way. Bin. There's no. Oh, here. That's what's printed out here. Okay, here. Okay, here we go. So now, just to repeat what's happening here, I've read the file and converted into a record object provided by PyMaxMyth. Now I'm going over interactions there. Okay, I'm printing for interaction type, and how many participants is there. So this is one case. We've got association between 11 things. Okay, and then I'm checking how many of those we've got. If there's more than two, I'm checking what's the interaction type. If it's association, then I'm expanding according to the spoke model. Okay, and once expansion is successful, the bin list is gonna have one or more, two or more actually expanded binary interactions. So I'm going over this list. I'm getting for each participant, I'm taking the label. I'm taking the label of the reference protein. So this is identifying the the protein we're dealing with. Okay, so I'm getting the label for one protein, for the other protein, and then I'm printing it out as you see here with this PP here. So this can be used as an input to the for the cytoscape. So this way we've got the script that takes interaction records from a file. Interaction records, parses them, gets the individual binary interactions or binaries resulted from expansion, and then creates a file that can be used as an input for Cytoscape. So that's how this script works. This is initial very simple version. At the moment we're using, oh, one thing to notice, this is a clearly a spoke expansion because the first protein here is always the same. We've got ESA1, so this was presumably bait, and then we had different praise here, and the same thing here, ESA1 and so forth. So there are apparently three experiments where in each of those ESA1 was acting as a bait. So this is spoke expansion clearly. So the module figures out which protein is a bait, which are praise, so it does it according to whatever the table was showing up. So that's the simple version of the script. It needs a little bit of work because we've got to get here, instead of the label, we've got to get the same identifiers as in the input file. So the script will have to be modified a little bit, but essentially it does whatever is needed to generate the interaction file for the cytoscape. So I'll stop here. I'll get the script to the version where it generates the proper identifiers. Okay, and I'll put the updated version of this script on the server, so you can try to play with it and use it as an input for the cytoscape. So that's how it goes. I'll also put a bunch of references to different bits and pieces that I was mentioning today, so that you can find the reference information and how things work. So that's about it. It's, I'm not sure if it's whatever you expected, because it's apparently heavily based on scripting and generating data for Cytoscape. So it might not be exactly what was advertised. I think there was some miscommunication, and the web page didn't reflect exactly the content. It's gonna get updated for the next quarter. So but hopefully it get you some motivation to go after Cytoscape with the intention of generating input files on your own with the scripts. As you see, it's not that hard. Being able to read the records from Uniprot and being able to read the interaction files returned by Psychic gets you a lot of input. It's not that complicated. Parsing XML is not hard, and this way you're gaining gaining control over what's getting into Cytoscape. So hopefully it's useful. If you do have any questions, comments, please send me email. If you want to if you want to start writing scripts for Cytoscape input, if you want to use Cytoscape, let me know, and I can try to help in whatever way. So feel free to email with me with questions and problems. If you do take the workshop for a credit, please drop me an email, identify yourself, and tell me if you're on a programming side or not, because then I'll try to get you an assignment which is gonna be either based on scripting, some simple scripting, or just on dealing with Cytoscape by clicking and doing things inside Cytoscape. So please let me know which side you are, so that you're not gonna run into problems doing things that you don't you're not interested in, or don't know what to do with them. So that's about it. Are there any questions? Unmute yourself and at least try. I can try to unmute you if you're blocked by any chance. I don't see anything in the chat. I don't see anything in participants. So I can ask to more. I can No, I can unmute. I can unmute. I can. So if anyone's got any questions, you should be able to ask on. You're not blocked. So if there's no questions, then thank you very much for patience, and please drop me email if you do have if you're on a if you're taking the workshop for the credit. Okay, so that's and once more, I today in a later during the day, I'll get you the updated version of the current Python file with the script and the pointers to different resources, so that you'll have something to start with. Otherwise, thank you very much, and thank you for participation. And I think that's about it. So I'm getting offline. I'll keep the session running. So if you got more questions, you can also drop them
Thank you.
drop you in a chat. So let's see. And I'm going